So thanks for having me. I'm going to start today by giving you a sort of perspective of where we're headed in trying to determine high-resolution structures of various types. And I'm going to do that with this graph, which very nicely illustrates the sort of progress of the types of machines that are used to determine structures. So these are, in particular, X-ray generation. Yeah, sure. I see. So these are generating X-rays. These are mostly synchrotron facilities. There are some X-ray free electron lasers here, which are extremely bright, and this is the year in which they've sort of come online. And what I also want to show associated with that is how much data is being generated by these facilities. And it's quite a bit of data for even the earliest facilities. It quickly rises to about 100 terabytes per day for some of the more advanced synchrotrons, which is a lot of data. That's equivalent to the global internet traffic in 1997, which is putting things into perspective. The newest facilities that are supposed to come online are going to be generating over an order of magnitude more than that, and that's equivalent to the global internet traffic in 2003. So we're scaling really fast here. And these are the types of machines that I'm talking about. So on the left are these synchrotron X-ray facilities that generate beams of X-rays that impinge on a sample, and you're detecting the types of interactions that happen as a result of that. There are even bigger facilities that are brighter, and these this nice line, which is almost a kilometer long, illustrates one of these lines. At the end of this is your sample, and it's being impinged by X-rays. The third type of experiment that I'll tell you about today is a bit different. It's sort of a tabletop experiment compared to the first two. And this uses an electron microscope to do the experiment instead of these. The biggest difference is that you can have one of these in your bedroom if you'd like, whereas you can't with these. And so I'll tell you first about experiments to do with cellular ultrastructure using synchrotron X-rays. And I'll start by saying, the concept of data reduction for X-ray crystallography is best summarized by looking at a picture of something like this. This is produced when a beam of X-rays is hitting your sample. Your sample is a crystal, and it produces an interference pattern, and that's what you're recording. This interference pattern produces a number of peaks that you see here. You see them arrayed across this detector, and then almost nothing or some background information around the peaks. And what you're interested in for the typical crystallography experiment are these peaks. And you try to count these very accurately. You try to figure out where exactly they are in relation to all of the other peaks. And then you try to integrate that peak as accurately as possible. And from that information, you can try to deduce what the integrated intensity of that peak is and what the error associated with that integrated intensity is. That's what's going to tell you your structure. What I'm not telling you is that you're actually missing half of the information to determine that structure, and there are methods that are used to achieve that. The first experiment that I'll tell you about differs profoundly from that case because whereas you could ignore a large percentage of the information that you were recording in the image that I showed you before, in this type of experiment, you're actually caring about every single photon that comes out of that interference event. So when this X-ray is interacting with your sample, every pixel matters. And so you have to use the entire collection of pixels in this whole image to try to retrieve the structure of your object. In this case, your object is a cell. You do this by implementing this type of algorithm. This is a representation of many different flavors of this same type of algorithm where you start with an initial guess for what your structure might be. That guess can be extremely general. Usually it's just a constraint on the size of your object, and that gives you an initial set of phases that are associated with each pixel in this image, and then you refine based on some constraints. You know, for example, that your object must be positive, and it has real positive density. You know a few other priors, and then you can try to come at a solution for your object. And you can do this relatively successfully. These are some examples of this type of approach, trying to render subcellular structures in 3D. So this is a yeast spore where you can see, for example, the nucleolus here, and you can see some associated structures with the cell. In other cases, you can see different types of cells have a different morphology, and that's been able to be captured here, and you can also see subcellular structures in there. The subtype of experiment of this category that I'll describe today is a scanning type of experiment. The primary difference between this first experiment and the second one is that in this experiment, to get a single two-dimensional projection of your object, you record a single pattern. So that's a single picture. In this case, to get a single two-dimensional projection of your object, you have to record thousands of overlapping patterns. So you scan the X-ray beam across your sample in a way that any given location overlaps by a certain percentage of the sample with the adjacent location. So you move over, not quite one beam profile over, and you use that overlap constraint to try to tell you something about your structure. And when you satisfy all of those overlap constraints, that gives you a single two-dimensional projection from all of these individual diffraction patterns. This is what a sort of close-up of what the experiment looks like. So this is your X-ray source coming in. It gets focused onto your sample and then diffracts in this way, and you can raster this stage across. This is what a typical sample looks like. So you have here some this type of grid-like structure, and there are cells on this grid. Here they've been consuming some kind of fluorescent nanoparticles, so you can see that the cells are labeled in red. That's the overall cell. And then within the cell, there are these accumulation of green particles. And that's what we're trying to track within the cell. This is the type of substrate they're on. This is a very X-ray transparent substrate. So essentially, the only thing you're going to be looking at is your cell on this fine mesh. And this is a type of procedure you have to undergo to actually retrieve the structure. So this is the structure that was finally retrieved. This is a leading edge of a cell that has engulfed some nanoparticles. And what you see on the right at each of these steps where the circle is scanning across the image is the actual diffraction pattern that was collected from that location in the sample, right? And you then have to use all of these diffraction patterns to come up with this particular image. This is only looking at a 3 by 10 micron piece of cell. It's been cropped here, but even that 3 to 10 micron piece of cell requires something like 10,000 patterns to be acquired. So this is a very high-resolution data set. You can actually see particles within this data set that have features that are less than 20 nanometers, and the entire cell itself is something like microns large. The penetration depth here is not an issue because these X-rays are at an energy that can go through at least about a micron of sample of this density, and so you can get very, very thick structures at very high resolution. And if you combine a bunch of these 2D projections now, you can assemble an entire tilt series so that you can go from about minus 60 to plus 60 degrees and use these for trying to reconstruct a tomographic projection of your object. The issue here is that each of these individual images requires something like 40 gigabytes of data. And the entire data set can easily be something like 8 terabytes of data. So trying to do this on the fly is a real challenge. What we do is we use an on-site GPU cluster that runs algorithms locally that tries to reconstruct these images. That can be done within 2 to 5 minutes. So you wait about 2 minutes after having taken a series of patterns for this particular projection, and then you tilt your sample and you collect another one. The data is then taken back, and then we do 3D reconstructions at home. We're trying to do this in a better way. So one of the students, in collaboration with John Miao's lab, has looked at trying to make a package so that you can essentially try to do this on the fly. So this is very fast tomographic reconstruction. It's meant to be a general type of a Fourier iterative reconstruction algorithm so that you can upload your data or give it a link to where your data is. And this runs on many cores or even on one core and tries to give you a 3D reconstruction within a reasonable amount of time. I'll leave that there, and I'll move to the second type of project that is even, in my opinion, more challenging than the first. It deals with higher resolution structures obtained from very, very small crystalline assemblies of proteins. There are some examples of these assemblies here where you can see nanocrystals here grown in bacterial cells. So these nanocrystals are no more than about a micron in size. Here are a few other examples. This is a yeast prion crystal that grows about a micron long but not very thick. And we want to try to get atomic structures from these. There are many other examples of these types of nanocrystals that may be present in typical crystallographic screens that are used to try to find bigger crystals. So hidden within these, there might be these nanocrystals. We use electron microscopes, much like this one, to try to determine the structures that I'm talking about for these molecules. The way we do this is we put the sample in this small hole that you see here. A beam of electrons is generated right up here and barrels down this column. It impinges on your sample. The diffraction occurs somewhere in here, and you record it in a detector somewhere below. This diagram represents that type of interaction. Here's the beam of electrons. This is your crystal, which then can tilt within the beam. At each orientation, you record a diffraction pattern. The whole thing amounts to having a movie, and from this diffraction movie, especially if you have a prior guess for what your sample might look like, you could try to derive a structure. This is a real diffraction pattern from one such protein crystal. So this is the protein crystal. This is a half a micron scale bar, so you can see that it's very small. And you can see that there are thousands of spots. So this is the typical type of problem I'm discussing when we're talking about sort of the canonical way of addressing crystallographic data reduction. This is just another schematic looking at the crystal top down. You can select from a small region of the crystal that you want to actually collect from. And this is one of the movies that you're looking at as you're tilting the beam. You're tilting your sample within the beam. This is the axis of rotation of your crystal, and you can see that spots flicker in and out of diffracting condition. And these spots are then integrated by the data reduction programs to try to achieve your structure. Those programs try to take a region like this and identify the spot, as I mentioned before. You can assume that represents a sphere of reflection in three-dimensional space being sections of it being excised and recorded by your detector at any given time. And as you run across the sphere, you record different wedges from this particular sphere. You can then add all of those up and come up with an entire profile. And the signal-to-noise ratio we're talking about is somewhere between 1.5 and 20. So that's pretty good, and that produces structures like this. So this is a structure of a small peptide segment that was determined completely ab initio, so the intensities that were recorded were good enough by themselves to tell you what the structure would look like. And you can do this for more complicated samples. So this is a prion protein, which is notoriously difficult to characterize. A segment from that gives also very high resolution features, and this is the type of structure you get out of that. So this is a molecular assembly that's extremely tight. There are actually two views of this, one of it without the hydrogen atoms and the other with the hydrogen atoms. So you can see that there's literally no space here. This becomes important later on because this essentially is a characterization of nearly every single atom in this structure. We know its exact position, and we know its exact species. And we can start to assess, biochemical features of these structures that might inform us about disease or some other aspect of the structure. And we can also use those in a second experiment, which is the point of this whole second bit, which is where we take a very, very small beam. The first electron beam was something like a micron in size. In this second experiment, we use a beam that's only 5 nanometers in size. So just to put this into perspective, one of those molecules that I showed you in that nice assembly was only about 3 nanometers long. So this is just slightly larger than one of the length of one of those molecules that I was showing you before. So now we're taking this same crystal and we're raster scanning this small beam across it, not in overlapping steps, but far enough away that one diffraction pattern does not interfere with the other. And so we do this over the entire crystal. We use this nice instrument here. So this is a slightly bigger electron microscope that can focus the electron beam to this very small probe size. And we're now detecting the entire diffraction pattern here. This is operating at 400 frames per second, but an upcoming upgrade will start recording at 100,000 frames per second each of these images is 4K by 4K at 16-bit depth. So this is going to be tremendous amounts of data that we have to go through. And the reason why it becomes more complicated is that we're no longer just integrating spots. The types of spots that we see here, because of this interesting geometry, look a little bit different. So they look almost like circles here. And within the spot, you actually have additional information other than just the absolute intensity. You have information about the substructure within the beam. So this is the type of movie that we would record. So what you see here now on the right-hand side is a digital map of where these spots are lighting up, represented spatially over a 2D projection of the crystal. So what I mean by that is we take these little circles, and each one of these little circles maps to a given panel. And where the spot lights up for that circle, you can see stuff lighting up in this map of the crystal. And what you can see is, once this loops over again, this produces not only a 2D map of the crystal, but it gives you a lot of morphological information for when these spots appear or disappear. So unlike a typical experiment that would sum all of this into a single diffraction pattern, now this is broken up into thousands of different patterns that give you nanometer scale orientation or information about the types of structures within your crystal. Here's another example that is a lot more heterogeneous. So in the first example, you could see the spots sort of either appear or don't in a single lattice. Here, the spots seem to sort of flicker in and out in what appears to be incoherent lattices, but you can see that if we sort of overlay what the expected lattice should be, they more or less match the expectation, but they don't all light up at the same time. And what this means is that there's a lot of heterogeneous structure within this crystal that is not captured by the average lattice, but is present when you start to look at the fine details here. Right now, we're playing this sort of game of trying to extract as much data as possible. And I think these are severe limitations because we are only going to be more inundated with data, and so we have to figure out better ways to deal with it. The first thing we do is we play the data hunger games, which is, you can only record so much data in a given time. So if something's not looking good, you just don't record the data. And that's not great. We should be able to record all data and then be able to have at least some kind of established metrics for what should be kept and what shouldn't be kept. There's no long-term storage. You're not able to just store terabytes of data somewhere in a national lab. So all of this is currently transferred, and there's some attempts to store it locally, but this will become more of a problem. And there's no on-the-fly processing. So this is recorded and then over a period of weeks processed later. And so better on-site processing is almost, in my opinion, a requirement if you want to be able to get more rapid information to inform your experiments. And this sort of brings us back into perspective. So this was the global internet traffic in 2003, and this is from these fancy experiments. This is a single electron scanning diffraction experiment where in the new type of geometry we'll be generating something like 10 petabytes of data per hour. And in that sort of setting, we're reaching the global internet traffic of 2007. So that's a lot of data. And it requires a lot of people to do this. So this is my lab, but we have extensive collaborations across campus. We collaborate with the Janelia Research Campus that's funded by Howard Hughes, and we have a bunch of national labs that are actively pursuing these problems because it's not just an issue for us, it's an issue for anybody else who's trying to do this in these shared resources. So with that, I thank you for your time, and I'll take any questions you might have. Yes.